This is to help you assemble your running system and load your samples into your gel. This is an example of a running system. This is the older one, which most of you will have. You have a piece inside, and this piece is where you're gonna put your gels. You can run 2 gels at once in this system, but in my case, I'm gonna run just 1 gel. So if you have an odd number of teams in a cubicle, at least one team will need a dump plate. This is the dump plate. If you read what's written in the middle, it will tell you which side should face the middle, what they call a gasket. So put your dump plate in the mid part of the gasket and remove your gel from your bag. Don't remove the comb yet. Place your plate with the comb facing the middle of the gasket, like that. Make sure both plates are all the way down to the gasket. Place one system inside of the other and close the clamps. You can see here I'm drying my plate because I'm gonna draw the wells before I remove the comb so that I can see it later when I place the buffer inside. You cannot see in the camera, but I will show you later. Okay, you can see all the wells at the very bottom. I made a little line so I know where they are. Place the gasket inside of the chamber. This will create a system where you're gonna have buffer in between the 2 plates and buffer outside. I'll pour the running buffer in between the 2 plates first. All the way to the top. And then I'm gonna pour buffer outside. I can now remove my comb. And as you can see, you can't see the wells pretty well. That's why I made the lines on the outside. I'm showing you here the samples we're gonna run into the gel. So I set up my volume and I'm gonna take my tip, and you're gonna have a special tip that's much thinner at the tip, and it can go easily in between the 2 glass plates. So I'm pipetting my first sample to my first well, placing the tip right in between the 2 glass plates into my well. And you can see the blue setting at the bottom of the well. Then you can proceed loading your next sample. In this case, you'll be loading your samples twice in the same gel. And that's what I'm showing here. Once the samples are loaded, then you can proceed to the lather. I'm gonna add 5 microliters of the lather that I'm showing here. After you're done loading all your samples, make sure the buffer in the inner chamber it's above the sample level because sometimes it leaks. So pour a bit more running buffer all the way to the top glass plates to make sure your buffer is touching your samples. And if you still think it's leaking, you can pour more buffer outside in a similar level from the inside, just to make sure you're gonna have a current going from inside chamber to the outside chamber. Close your running system with the black electrode on the black side and the red electrode on the red side, and plug it properly on your power pack. You can now set up the recommended running sets from your lab manual and start your run. You will know everything is going right if you look at the chamber and you see the bubbles going up. And you'll also be able to see the blue starting to go down into the wells. Here is after a few minutes and you see the blue starting to go down. And here is almost at the end. I usually like to let the blue reach the green rubber at the end, and that's when I decide to stop my run to make sure none of my protein bands will get out of the gel into the buffer. But depending on the size of your protein of interest, you might want to let it run longer, or you might even stop earlier. It will all depend on your sample. I'm stopping my run right now. I'm removing the lid, and I will slowly lift the gasket, the inside chamber, from the outside to make sure I'll remove all of the buffer and not make a big mess. Pouring the buffer inside. You can throw away that buffer in the sink. And you can open your clamps and carefully remove your plate. You can see the blue front at the very end of the glass plate. You can get the green lever that will help you separate the two glass plates. You can separate them, and the gel will remain glued to one of the two plates. Now is the time you're gonna use the same lever to remove the stacking gel. You can throw the stacking gel away. Because you're only gonna work with the resolver where your proteins are. In this case, I'm taking half the gel for the gel dock and the other half I'm using for a Western blot. So I'm gonna use the lever that I used to separate the glass plates to split the gel in two right after the ladder. And then you choose one gel to do your transfer, and the other one you're gonna look at the gel dock. That's your SDS page. You can see I'm very careful not to break the gel. And you can use a bit of water to make sure the gel will slide nicely on the drawer for the gel dock. Since the gel dock takes a long time to read, you might consider putting all your gels from your cubicle at once on the drawer to acquire the image all at once, because it takes a long time. And now you can take your drawer to the gel dock and capture your image.